All right, good morning, everyone. Thank you for coming for day 2. It is my pleasure to introduce Andy Hoofnagle. He is an MD, PhD at the University of Washington, and in Boston vernacular, he is wicked smat when it comes to doing targeted quantitation. And I've enjoyed his presentations and interacting with him over the years. Andy, take it away.
Thank you. So I help run the clinical labs out of the University of Washington. Can everybody hear me okay? It's hard to know if my voice is making it out there. Is it okay? Up a little bit? I'll try. Do that again if I'm getting quiet, okay? So I help run the clinical lab. Today I'm going to talk about calibration first, and then some of the basic science that we're doing in HDL proteomics. That's going to be a theme throughout. But I am going to talk about a couple of the assays that we've launched and actually run clinically today. And so stop me as I'm talking. I would hope that I'm able to tell you about what's going on in our lab, but please feel free to ask questions because it's better that way. So to start out with, I'm going to tell you a little bit about HDL proteomics and provide some of the data that we've gathered in our lab that hopefully will make you realize that calibration is something that you can do and that you should do. It's not hard to do, and you should do it, and it will help your results within your lab, but it will also help people who want to replicate your work in other labs. So why talk about HDL proteomics? The reason is lying in this page. This has been known for a very long time that the lower your LDL cholesterol, the lower risk we have of coronary artery disease. But the more HDL cholesterol you have, the lower risk of coronary artery disease that you have. So no matter how good you are at getting your LDL cholesterol low, if your HDL cholesterol is low, you're at increased risk of cardiovascular disease. So HDL has been called the good cholesterol. But it can't be the cholesterol that's the entire story. So there are a couple of trials of HDL raising treatments. So these are CETP inhibitors, torcetrapib and dalcetrapib. 2 very large studies demonstrated we can raise HDL cholesterol. Everybody thought this was going to be amazing, fantastic, we're going to save lives. And instead, we had an increase in poor outcomes in Illuminate and no improvement in outcomes in the DAL outcome study. Niacin is another way to raise HDL cholesterol. People don't like it. You take a lot of niacin, you get flushed and things. But you can raise your HDL cholesterol, and the aim high, we had twice as many strokes. So all of these studies were considered total failures. And that proved to us that pharmacologically raising HDL cholesterol may not be the best thing to do. When we think about what HDL does, it's a particle. This is Apolipoprotein A1. I'll be talking about that a few times in this morning. Apolipoprotein A1 can interact with lipid-laden macrophages, which are the fatty streaks and the atherosclerotic plaques that we have in our coronary arteries. ApoA-I can come in, grab lipids from those lipid-laden macrophages, and take via HDL to the liver for disposal and excretion in the feces. Apolipoprotein A1 can also, and other proteins, can interact with neutrophils and prevent their degranulation. So it's anti-inflammatory. In addition, there are proteins in HDL that are known to inhibit the complement cascade. Lastly, there are proteins that can inhibit the peroxidation of different lipids in HDL and other lipoprotein particles. So it's antioxidant. So while HDL cholesterol is the marker of good health, in fact, it's all of these other functions that are thought to prevent coronary artery disease more mechanistically. And this was actually borne out in a paper from Dan Rader's lab, who's at the University of Pennsylvania. And this paper from New England Journal of Medicine showed that you could use HDL cholesterol to pull cholesterol from lipid-laden macrophages in a test tube. And as we looked across the large community-dwelling population, you can see that if you have diabetes and hypertension, you're more likely to have coronary artery disease. Smoking in this study wasn't statistically significantly associated with outcomes. LDL cholesterol, neutral. HDL cholesterol by itself, not statistically significant. It was the ability of those HDL particles to pull those lipids out of the macrophages that was associated with coronary artery disease. So it suggested that it's actually the function of the HDL particles, not the HDL cholesterol itself, that was helpful in preventing coronary artery disease. So when I was doing some of my postdoctoral work in Jay Heineke's lab along with Tomasz Weiser, we developed a procedure to quantify proteins in HDL. So using density gradient ultracentrifugation, we can actually tease apart the low-density lipoproteins from the high-density lipoproteins. And then we can measure what proteins are in the HDL to try to figure out if we can learn something about HDL function in different disease states. Using shotgun proteomics, we were able to demonstrate that there are a lot more proteins in HDL than we ever thought there were, and this list has been expanding ever since this paper was published more than 10 years ago. But some of the interesting things that we found in HDL were protease inhibitors, which I'll show you on the next slide why that's important, and regulators of complement. And the two combined, when we think about what actually causes a heart attack, a myocardial infarction, we have the atherosclerotic plaque, we have the fibrous cap on top, enzymes, collagenases and others, weaken that fibrous cap. Now we have protease inhibitors in HDL that are preventing that from happening, perhaps. And we have this platelet and coagulation and activation when the lipid core gets exposed as that enzymatic weakening of the fibrous cap. We have exposure of this core, we have clotting, we have occlusion, and we have cell death, like myocardial infarction. So it's thought that perhaps with these protease inhibitors and complement regulators, perhaps we can prevent the formation of these clots, and maybe we can have better outcomes because of that. So that was some of the hypotheses that we had. So what I'm going to do now is talk about different ways that we can use proteomics to quantify proteins. So first, let me tell you about the study. This was something called the egg feeding study in Seattle. This was 30 normal people. Well, so this study was, well, we call them normal. So they had to eat 4 scrambled eggs a day for a month. Then the next month they ate 2 scrambled eggs and 2 egg beaters, and then the last month they ate 4 egg beaters a day. That's disgusting. So that's normal, though. They were normal. They didn't have anything else wrong with them, but they did sign up for this study, which was questionable. But anyway, that has absolutely nothing to do with this. This was actually all the baseline samples that we're looking at, just to learn something about the distribution of proteins in the normal human condition. So we're not looking at crazy disease states. This is just trying to quantify proteins in HDL in normal people. And so we're going to compare different ways to quantify proteins. Looking at shotgun proteomics, we can use spectral counting and extracted ion chromatograms. And then in targeted proteomics, we're going to look just at peak areas using transitions, 2 peptides per protein, including isotope-labeled peptides, which, remember, that can get kind of expensive, right? Unless you're looking at crude, we were using purified. And then finally, looking at a single isotope-labeled protein, ApoA-I, which is the most abundant protein in high-density lipoprotein particles. So on the y-axis of each of these figures is an immunoassay measurement of each of these proteins. I don't like immunoassays, and I can talk for a long time about how much I hate them. I'll bring that up in a little bit. But everybody in the clinical lab knows that immunoassays are what we use clinically, so they want to see these kinds of data. So just comparing immunoassay for each, these are just 6 proteins that we're looking at in HDL. They were the immunoassays that we had available. You can see that spectral counting doesn't really replicate this biochemical assessment of the protein concentration very well. So, because we were on a high-resolution Orbitrap, we decided to just, okay, now instead of spectral counting, we'll look at extracted ion chromatograms. Things didn't do much better, which was surprising to us. We thought that we would be improving the specificity of the measurement by using that approach. When we went to a triple quad and started to do transitions specific for each of these peptides, it also didn't look much better, which again surprised us. But when we started to include internal standards, things got much better. So the correlation now between the MRM with isotope dilution compared to immunoassay started to look significantly better. This is 12 peptides, and if each of those are $500, we're now up to significant amounts of money to just provide internal standard peptides. We said, well, what if we can use an internal standard protein? So this is an isotope-labeled Apolipoprotein A1. Every nitrogen is labeled with N15. Can we use that as a surrogate for internal standardization? And the answer is yes. So if you look at the Apolipoprotein A1, it's going from 0.79 to 0.96, so significantly improving that, but that is the internal standard protein of interest. We can use surrogate peptides from ApoA-I as internal standards for other peptides in these other proteins and see a significant improvement over just MRM alone. So internal standards, because of those matrix effects and because of the digestion differences in sample to sample, internal standards can really help a significant amount in normal human biology. When you have very disparate disease states, it may not be quite as important to determine a difference between disease and not disease. So we went in just being a lab medicine kind of guy. We wanted to see where is the variability from this assay, and is it linear? Because if an assay is linear and precise, and we can replicate that over many days, we actually have an assay that means something, and other people can do it. So we took a controlled HDL. So here, this is a normal human donor pool. So many plasma samples that were pooled together. The HDL was purified from that. We're going to do 12 digests all in one go, pool them together, and inject that gazillions of times. This is going to give us an estimate of what the variability at the instrument is at the LC-MS step. And then if we take 5 digests and not pool them and inject each of them 4 times, we'll be able to remove the variability from the LC-MS part and get an estimate of how much variability there is in the digestion. So this kind of pool split experiment is a way to tease out the variability in your experimental design. We then wanted to compare internal standard peptide versus internal standard protein. So when we look at the variability of just the LC-MS step, the internal standard peptide does a nice job of reducing the variability. Again, because of that experimental design, we can look at what the variability of the digestion is, and then when we use sum of squares or an ANOVA approach, we can combine that together to get an estimate of the total variability in the assay using an internal standard peptide. Expensive. Using a single internal standard protein, we can actually reduce the digestion variability, right? Because the trypsin is now cleaving up the internal standard to make the internal standard available for the LC-MS. We have a higher variability in the LC-MS step when we use an internal standard protein, but when we combine it together and look at the total variability estimate over time, things are actually pretty similar. So we can use a single protein in an experiment like this to reduce the variability to digest an LC-MS variability and save significant amounts of money compared to internal standard peptides. But it doesn't work all the time, and it's actually works less well for other proteins. We got lucky with these 6, so you need to validate that in your own system. So linearity, we were able to purify mouse HDL and dilute our human HDL with mouse HDL and show that the measurements of each of these 6 were linear, both with peptide and with one internal standard protein. Okay, so any questions about that? That's the basis of the HDL proteomics part. Please.
Just real quick, is there any difference between pooling those samples versus just having an individual's HDL sample versus pooling? From an analytical point of view, I mean, I think of them as the same, but I wonder if somebody could argue that there would be some sort of difference when you...
It's a great question. So so if you pool together about 400 people, you'll basically get rid of variability from person to person, right? So, and if you make another pool of 400 people, it'll look almost the same as that previous pool of 400. This is not 400 people, so we already have individual variability kind of included in a smaller pool like this. But by pooling, we remove the between individual variability. We have no assessment of that. If there is individual protein, for instance, in me, that's significantly lower than you, then the variability measurement will be different. So the answer to your question is yes, it's different, but it's different because of the protein concentration and possible matrix effects.
Okay, but from an analytical point of view, I mean, would you just say, moles of analyte in this sample is, versus an individual sample? I mean, that's kind of, having it be pooled versus an individual didn't really...
The pool versus an individual, yeah, it doesn't affect our ability, our assessment of the variability to a great extent. It does do different things for your epidemiological studies later, whether you pool or not pool. Yeah, but we don't pool. We like individual samples for the most part. This pooling experiment was just so we had enough sample to carry it through 90 injections. Does that make sense? Yeah. Any other questions? Yep.
How often you run the pool sample for the LC-MS variability testing?
How often do we run it? We run it with it depends on the experiment, but we prepare, for instance, for this, we're preparing a control sample in the HDL step, then we have a pooled HDL that we're doing in the digest step, and then we have a previously digested sample that we're using for system suitability. So we have quality control materials built in at each step. Does that make sense? So it depends. System suitability is usually every 10 to 12 runs, whereas the batch and digestion, the HDL purification and digest parts are per digest or per HDL run.
And is the peptide still stable over a period?
Is it, let's say that?
Peptides are very stable still over a period.
I don't know if we've talked about peptide selection. Did you guys talk about that yesterday? But not peptide selection, not so much. We try to pick peptides that are stable. So one of the tricks that came out of the McCoss lab is to just leave your sample sitting on the instrument and inject it 72 hours later and use DIA approaches to see what peptides are stable or not. We've worked with all of these peptides for so long that we've injected the same vial over and over again. We know what peptides are stable, but that's one of the most important things to look at is how stable your peptide is on the instrument. It's a very good question.
All of these validation experiments that he was describing in more detail in each one of the sessions today.
Excellent. So I'm going to table that question until later. Any other questions? Okay, so I'm not going to talk about the selection of peptides because we're going to talk about that later, but I do want to talk about digestion and how painful it is because that's the motivation for doing the calibration, okay? And then, so we've talked a little bit about internal standards already. I'm not going to go into quality control in any great detail, but then I want to talk about comparing results between laboratories because that's where calibration has really helped a lot. So when we think about internal standards, these are M15 labeled, so they're co-eluting almost identically. If you have deuterium, you'll have a little bit of a shift. This is controlling for LCMS variability, or if we actually have an internal standard protein for digestion variability. And just this ratio of the peak areas, endogenous to internal standard, which we're going to call the peak area ratio or the response. The question is, how variable is the response? If I take a sample, a digested sample that I digested in Seattle, and I run it on my instrument, and then I bring it to Boston, and I give it to Sue, and I put it on her triple quad, how similar are those two peak area ratios going to be? Any guesses? So they published a paper, this experiment, this is, Sue, you were on this paper too, right? This is, yeah. So this, I'm not going to go into detail about each of these experiments, but this is the simplest thing, what I just described. Digest a sample locally and send it out to a bunch of different labs and find out what the variability is. And you can see that just the variability of the peak area ratio ranges, 4% is pretty good, but we see some peak area ratios varying as much as 13.7%. That was shocking to me. I wasn't ready for that. This has been replicated over and over and over again. That a peak area ratio is not consistent but from lab to lab. Something to keep in mind. And then, if we actually include the digestion into that and find out how variable measurements can be, again, just looking at the peak area ratio, the variability of taking a single digest and injecting it once, these measurements, again, from lab to lab, can be extremely variable. One of these is greater than 30%, which means I can't tell the difference from 0 versus the measurement. This is just something to keep in mind. So why? Why is digestion so painful? Why can't we get it right? Part of it is you have so many options. That's what's frustrating about proteomics is there's no cookbook for any of this stuff. Are you going to pick urea, Rapigest, TFE, acetonitrile, lots of other detergents? Are you going to reduce or not? Are you going to use DTT or TSEP? Are you going to alkylate or not? Are you going to quench your alkylating agent after you're done alkylating? Are you going to use trypsin with Lyc or without Lyc? Are you going to switch to Glu-C, which is a new fun enzyme that we're using in our laboratory more and more? And then what is the pH? What is the pH after you added your trypsin? Is it less than 7.8 or is it 8.1? And I'm going to talk a little bit about mixing and how long we incubate our samples for. There's so many steps that you have to think about and try to keep optimized that is kind of bewildering. So these are some data just from Apolipoprotein A and Apolipoprotein B, which I'll be talking about a little bit more. So if you use TFE versus urea, you can see that the peak area ratio, the response, this is the response or the peak area ratio is different depending on which denaturant you use. Something to keep in mind. This was fascinating. So we are incubating this digest either in a thermomixer or end over end. Who uses it? How many people have done digestions? Is everybody good? Are we using thermomixers? How many people are using thermomixers? And who's doing end over end? Nobody's doing end over end. Okay, good. Well, one time Jess went home and the thermomixer stopped shaking. She doesn't know when, but when she got in the next morning, it wasn't shaking anymore. So she said, crap. So she repeated her experiment and it actually shook overnight properly. And then she came back and the relationship between two peptides within apolipoprotein A1 were different. Got a different result, different measurement. So then she said, well, this is kind of interesting. So then we have a lab quake end over end thing at 37 degrees. So she did it again. And it was a little bit different. The relationship between the two peptides was different based on how she mixed her sample. That's kind of scary, because which peptide am I going to use for concentration? Which one's right? So that was Apolipoprotein A1, which of course was a little bit surprising. But here is a different protein in exactly the same experiment. These two peptides are completely different from each other, depending on how we mix the sample. So just mixing, and whether you mixed for an hour, or mixed for 9 hours, or 16 hours, whether you did it end over end, or whether you were shaking, you could get completely different results depending on the protein. Something to keep in mind. We know that when you have to stop your digest sometime. Well, how long are you going to digest for? So some peptides appear and then disappear. This is because trypsin likes to start digesting things non-discriminately after it's had a chance to do some digesting. Here's one that starts out slow, speeds up, and then slows down again. So even at 22 hours, some of these peptides haven't quite finished their digesting. Others are completely stable. Jess decided to do, Jess Becker is somebody in my lab, so I'll keep mentioning her. She did almost all of these experiments. So what we decided to do is, well, what happens if you change the trypsin concentration? So this is actually on a log scale on the bottom. This is what the normal concentration of trypsin. It's half as much trypsin as you're supposed to add, and twice as much trypsin as you're supposed to add. And depending on the peptide, so these are different peptides from different proteins, one of the peptides, by doubling the amount, or this would have been quadrupling the amount of trypsin, we got back 5 times as much peptide as we would have if we didn't change the amount of trypsin. So now we have how long we incubate for, whether or not we mix, and then how much trypsin we added. Lots of ways to provide variability in your assay. Fun, right? Yeah, good luck getting that right? Okay, so that's why we want to calibrate. And again, it's not hard, but it's really important. So I've talked a little bit about Apolipoprotein A1 and ApoB. These proteins are totally different. This is a little 30 kilodalton protein, doesn't have any disulfide bonds, doesn't have any post-translational modifications. ApoB, which is actually the scaffolding protein for LDL, which is also called the bad cholesterol. This thing's gigantic. 250 kilodaltons with lots of post-translational modifications. It's about 10% sugar and many, many disulfide bonds. So completely different. And the question is, can we quantify both of these proteins at the same time? And is calibration going to help reduce variability over time? Why would you want to measure ApoB and ApoA-I at the same time? Well, ApoB is a better risk predictor than LDL cholesterol, and it turns out that the ratio may be even better. It's a little bit better in women than it is in men. So, how do you calibrate an assay? We want to turn that peak area ratio into a concentration. And Brenda was already talking a little bit about calibration this morning. There's this simple ratio of just taking the endogenous peak area, dividing it by the internal standard, and multiplying it by the concentration of the internal standard, whether that's at the protein level or the peptide level. Okay, that's the simplest thing that we can do. You can also get a little fancy. You can have external calibration curves where you take purified protein and spike it into an appropriate matrix. For instance, spiking PSA into female plasma, right? Female plasma has no PSA. Spike PSA, get yourself a bunch of different concentrations and use that to calibrate the signal into a concentration. And then what I'm going to talk about here over the next many slides is using an unadulterated human sample, whether it's plasma or serum, because that's what I do, but you could also use your cell culture, you could use your tissue. There's lots of things that you can use to calibrate, where we're using the endogenous protein over time to help calibrate our assay. So the hypothesis is that by using external calibration curves, we can reduce the between-day variability of the digestion compared to the simple ratio. And this is why I strongly recommend not doing this. The assumptions that we're making that the simple ratio of endogenous peak area to internal standard, we're going to say, if I do this little calculation, peak area of endogenous divided by internal standard peak area times the concentration of the internal standard to give us a concentration of the endogenous proteins, we make 3 assumptions. First, that the digestion is complete. Second, that the response ratio would be constant from digestion to digestion, or from instrument to instrument, which we already know is not right. And that the response is linear. Fair? Fair? Promise? I think this is fair. Okay, so those three assumptions are important if this simple ratio is going to mean anything. Okay, so now I'm going to bash those assumptions. Remember, just simple division, peak area divided by peak area. First, the digestion has to be complete. When is it complete? When, on this slide, is the digestion complete? Depends on which peptide we're looking at, perhaps, but I would argue you never know that it's complete, and from our data, you can never get serum to completely digest. It's impossible. Run a gel of your next trypsin digest, whether it's serum or tissue or anything. There's lots of bands there still. You might nibble out your protein, but you're not going to destroy every protein in the sample. Digestion complete. Depends on how much trypsin you add. If I add more trypsin, sometimes peptides get liberated better. I add more trypsin, other peptides disappear. When's it complete? How did you mix the sample? Right? Am I complete? Am I not complete? This is something that we never think about, but it's absolutely true. Response, peak area ratio, as you increase the concentration, and it depends on which peptide you're looking at and what your internal standard actually is, but you will see a power function. You'll see a loss of linearity as you increase the endogenous peak area. That's normal. That's supposed to happen. And if we plot it on a log-log scale, which lots of people like to do, right? Log-log scales make things look really nice. But if you look at the exponent, that exponent should be 1. If it's not 1, it's not linear. No one lists that equation very often. And actually, sorry, that exponent is the same thing as the slope of the relationship between response and analyte concentration. So this is something that we published, showing that the relationship between concentration and response is not linear. And this is actually a paper from Steve Har's lab. This is these dots, these symbols are where the line should be, and you can see that it deviates from a slope of 1. These are not linear relationships. So all three assumptions are gone. I don't know if my digestion is complete. It most certainly isn't linear, and as I've already described, getting a consistent peak area ratio from day to day or from lab to lab is almost impossible. Questions about that? So please, please, please don't publish peak area ratios times internal standard concentration. Leave it out of your papers. Don't do it. It's not worth it. You're not right, so don't say it. Okay. What about external calibration with the spiked matrix? We make a couple of assumptions here too. First, that the spiked protein is going to be similar in shape and post-translational modifications to the analyte of interest. That the digestion of those spiked proteins is similar to the native proteins, and that the relative slopes of the calibration curves should be similar across the days. Is that fair? You okay with that? Are these fair assumptions? No one's nodding. Does this make sense? So if I'm going to try to use a calibration system to turn my peak area response into a concentration, then the proteins that I'm using need to behave similar to the ones that I'm trying to measure in the sample. Fair? Okay. So we tried this with ApoA-I and ApoB. And what we found was on different days, the response, the peak area ratio, was different. This is versus an immunoassay from day to day. And importantly, it was different for ApoB versus ApoA-I. Again, this is day-to-day digestion variability. We could make a calibration curve. Beautiful, right? We can take bovine serum, doesn't have any ApoA-I, doesn't have any human ApoA-I or human ApoB, and we can spike in purified ApoA-I, we can spike in purified ApoB, and we can see, all right, cool, got a calibration curve. I'm going to use this to now calibrate my assay. So I have 2 days that I did the experiment, and the recovery that I got for day 1 on Apolipoprotein A1, pretty good, 89%, but on the next day it was 210%. I was twofold wrong. So, same thing, I had a calibration curve for it. Oh my god, I was 5 times wrong. So even with a calibration curve, with these purified proteins, from day to day, I wasn't getting the reduction in the variability that I was hoping to get, which was very frustrating to us. There are other things that we've learned. So this is an example of spiking thyroglobulin into bovine serum, and we can watch the thyroglobulin disappear from my calibrator over time. I have no idea why. Why would a spike protein disappear from plasma? I can't find it by mass spectrometry. Mass spectrometry, I'm supposed to be able to digest everything into peptides and find it. My protein was disappearing. You have a question. Going back. This one here. This is days. Sorry, this is days. And this is the amount of thyroglobulin peptide that I'm observing in our digest. And it's disappearing. So spiked protein is disappearing. But if I do the same thing, if I take a human sample and I digest it over days, the thyroglobulin doesn't disappear. So the spiked protein is disappearing, whereas the endogenous protein doesn't. Back to the assumption of the stuff I'm spiking in is supposed to behave like my protein, my endogenous protein. And this is an example where it's not working. You had a question?
You said this was going into bovine serum?
Yes.
And it's stocked solution, not yet digested.
Correct.
So it's something about the protease activity of the bovine serum being...
Yeah, but I should have my peptides should still be there, right?
They're not tryptic, you won't see them.
Sure. So if I have a protease that clips right in the middle of both of my peptides, because I have 2 peptides, then that would explain it disappearing. That's true. We've shown, though, if you leave it in the refrigerator, it doesn't disappear. It's something about this freezing thing, and I don't know what it is. It's almost like it's cold aggregation. Something weird.
That when we archive samples in the freezer, we'll, like even digested peptides resuspended in initial conditions in the freezer, we'll go back and reanalyze them and we'll see a drop in signal because there's something about the freeze thaw of the peptides that they just don't survive well.
So I think that Lindsay's going to talk about the freeze thaw experiments to make sure that your peptides don't disappear. We don't see this ever. Except this stupid experiment. It's not even an experiment. We were hoping that this would be the way that we calibrate our assay, and it didn't work. And I like your idea, proteases, whether there's some weird aggregation and not able to actually get the trypsin in. I really have no idea, but it was so bizarre. So anyway, the point is that this spike protein wasn't behaving like my endogenous protein, which was frustrating. So this is what I want to leave everybody with today, and I'm going to show data to prove that it works. So if we just take a plasma sample, I'm going to give you the example of assign the concentration with a clinical immunoassay because we're using immunoassays already to take care of patients. So if I say this concentration is 14, then that's a concentration that I can use. Brendan. This back here, that was unlabeled thyroglobulin. Totally bizarre. We spiked it into bovine serum.
Bovine serum.
Yeah.
If you go measure the same thing in serum,
it doesn't happen. So I can spike the same thyroglobulin into human serum, and it's fine. That gets back to the protease question. But very interesting. Okay, so, or what if I just look at a sample on the bench and I say 100 or 1, right? So take your favorite cell culture, put it on the bench, and say the concentration of this is 10. Okay, just make something up. Just make up a number. But you're right? You get to be right because you decided what was right. Okay, so however I assign the concentration, I'm going to do it with a clinical immunoassay. You do it however you want, but pick a sample that you say you know the concentration of. I propose 10, 100, some number like that. You could pick some other number if you want. Measure that sample in triplicate in each batch and use it as a single-point calibrator, which is a function in Skyline. So it's easy to do. It's cheating, but the hypothesis that we had was it could work better than some of the other alternatives. And that's what happened here. So I showed you that calibration curve, the beautiful calibration curve, where I spiked ApoA-I and ApoB into bovine serum, and I got bizarre behavior from day to day. But when we used a single native human serum sample and measured ApoA-I and ApoB in that sample and called it, we assigned the concentration using the nephelometric immunoassay, we can now quantify ApoA-I and ApoB in human serum and plasma over time and get the right answer, which was very exciting. So these measurements are spread over 3 days. So, and by using that native plasma as the calibrator, the within-day variability less than 7%, but now the between-day variability less than 12% for both peptides, using that the approach that I've just described. Okay, so that's results within one laboratory. We then had a hypothesis. I'm going to switch and stop talking about ApoA-I and ApoB and switch to talk about IGF-1. This is a method that's now being used to monitor for doping in sports. It was used at the Olympics over in Korea. So this assay is now being used and deployed in anti-doping labs around the world. So I wanted to tell a little story about how we decided to do calibration in this group as well. I said that I hate immunoassays. Here's a good example of why. So this is an IGF-1 assay on the Imulite 1000. It's a Siemens instrument. This is Los Angeles, and this is London. It's exactly the same assay on exactly the same manufacturer's instrument. It's exactly the same reagents. It's just run in 2 different cities. And this is the correlation of the samples. It's almost like it's a completely different assay. And the slope, it's 40% wrong at one site versus the other. Okay, so that's why I hate immunoassays, and that's why I love mass spectrometry. So IGF-1 is a short protein. It's only 7.5 kilodaltons. It's used in anti-doping, as I described. It also is used in clinical laboratories. For the analytical process here, we're going to do bottom-up. It's a small protein. Some could argue to try to do it top-down. But we decided to do it bottom-up, and we're using an N15 labeled version of IGF-1, and we're using a standard reference material. This is it's been assigned using amino acid analysis from the reference material manufacturer in Europe. So we wanted to figure out, first of all, what is the variability of this assay? And then we're going to ask the question again, can calibration help remove the variability of day-to-day? But first, let me talk about some of these experiments that we did. Or just think about where the variability could come from, going from lab to lab. First, we're going to take a vial of standard reference material from the freezer. So first of all, there's variability in picking which vial, right? Because some vials are going to have more protein than others, just be the way that we fill up the vials with the reference material. We're going to add some liquid and swirl the vial. We're going to make calibrators. We're going to pipette the sample, so there's variability just associated with the proteomic step, how much internal standard we added, the digestion, and then finally there's this variability associated with the LCMS. We wanted to figure out what is the variability of each of these steps, and can calibration actually improve the variability from lab to lab. We had an anally written standard operating procedure. This is somebody just suggested my lab wrote this. It's ridiculous. So she was careful to include both the American catalog number as well as the European, and she used words like reverse pipetting. Who knows what reverse pipetting is? We have one person. It's one of those things that you should learn. It's one of the most accurate ways to do pipetting, and it can help with various parts of your standard operating procedure. So so we had 5 laboratories spread around the world. Everybody had a different HPLC. Everybody did their gradient however they wanted. They picked their own columns, different mass spectrometers. Nobody was told what to do. The only thing that they were told to do was to measure 2 peptides from this digest. Follow the standard operating procedure, but do your LCMS however you want. Using that, we sent around 4 samples. We made digested the samples and the calibrators in-house and sent them out. The variability, we talked about the variability of the ratio, of just the peak area ratio. Using this calibration system, just the variability associated with the LCMS in 5 different laboratories was 5.6%. Pretty good, but higher than I thought it was going to be. I thought we were going to do it better. What about that step of making the calibration curve? What's the imprecision if I make a calibrator centrally and distribute it, or if I ask everybody to make the calibrator in their own laboratory? That's a big difference. So there's a lot of variability just in pipetting out the different concentrations of the calibrator into the calibration curve. Having a centrally made calibrator, the imprecision is just about the same as what we saw in the previous experiment. What's the variability of how much protein is actually in the vial? So NIBSC, this is the WHO International Standard, they actually tell you what their uncertainty is and how much they think they got into each ampule. And so the CV of that is 4.5%. Again, higher than I thought it was going to be, but not bad. So then we wanted to figure out what is the variability associated with making individual curves. We know the variability of what's in each of different vials, but so on different days we're making a calibration curve. We can get day-to-day variability. We can get making a curve on each day. So we can figure out. Every day we had 3 different calibration curves coming from 3 different SRM vials. We had 7 samples. We can calculate the overall CV. We can figure out from the package insert how much IGF is in each of the vials, 4.5% CV. We can actually then, using the within vial, how much variability is there making a curve, we can actually figure out what is the variability of just swirling the vial. It turns out to be 5.7%, more than I expected. Literally just adding sample, adding 150 microliters to the vial, and swirling it. There's 5.7% variability in that. So when we did this, we used the calibration curve. We measured 150 samples in each of the 5 laboratories. The average CV was 16%, much higher than what we were hoping to see. This was using a calibration curve. When we did it instead, so we had a hypothesis that if we used a single point calibrator, we would get rid of this variability. So we observed 16% overall variability. The hypothesis was by using a single point calibrator, it would be significantly reduced, and that's exactly what we saw. So using that single point calibrator, and again, we said what the concentration was. It's 358. You can now buy it. You can buy the single point calibrator where the IGF-1 concentration is defined. Using that approach, we can reduce the variability significantly across the laboratories. You can do this too. You do this in your own lab. Every time you do an experiment, have the same sample over and over and over again.
So
Please.
it is a single point external calibration.
External calibration.
That's
Single point external calibration. I'm using an external calibrator in your fancy algorithm, and I'm dividing everything by the peak area ratio of the internal standard calibrator, sorry, the external calibrator, and then I'm multiplying it by the concentration of that calibrator.
There's a lot of people refer to that sort of just taking the ratio that we talked about as single point calibration and single point internal calibration.
Yes, internal calibration is what I'm trying not to have you do. Does everybody understand his question? Please don't do internal calibration. Please, I beg you. Not yet. We're going to get better at it, but you can't do it yet. I don't know what time it is. Okay. So the last brief story is about thyroglobulin. Another reason that I hate immunoassays is that, so this is the automated immunoassay that we do in our laboratory for thyroglobulin. So this is a paramagnetic bead that's coated in streptavidin. It grabs onto the biotin on a capture antibody, which makes a sandwich with the thyroglobulin and a reporter antibody. So if I pull the magnetic beads and I look for all the enzyme that we grab onto, a flash of light means that a successful sandwich was made. Patients with thyroid cancer make autoantibodies, and those autoantibodies actually coat the thyroglobulin and they prevent the formation of a sandwich. So if you have autoantibodies and I give you a 0, you have to say, are you lying? And I don't know the answer. So these autoantibodies can interfere and give false negative results to patients who have autoantibodies, which basically makes us tell the physicians, go do something else because this assay doesn't work. So we started this about 14 years ago. We decided to try to use LC-MS to quantify thyroglobulin. And so the workflow is to digest the plasma sample, to spike in an internal standard peptide, so this is a heavy labeled peptide, incubate it with beads that has anti-peptide antibodies, elute off the peptides, and quantify it by LC-MS. Lee Anderson calls this cis-kappa. I call it immunoaffinity enrichment after trypsin digestion. The CPTAC made an antibody, so the CPTAC program, I'm not sure if anybody's familiar with it, but they made an antibody that's incredible. It has a very high affinity for the peptide that we're interested in. 3 labs are using, actually there's another lab that's not listed here up in Canada, and Quest has its own in-house polyclonal antibody as well. Everybody's doing it different. They're digesting differently. In this case, they're using a different antibody, and they're actually using different calibrators. So the native human calibrator at UW is me, and we assign the concentration based on the immunoassay, and then the BCR 457 is a standard reference material. These others are calibrators that are available from a manufacturer. This is how good the immunoassay is. This is 4 immunoassays being run in the same lab at Mayo. You can see that the concentration is twofold different, up to twofold different. That's a lot. But when we do it by LC-MS, 3 of the laboratories essentially overlay with one another. Fantastic. So we were able to take now LC-MS, a good antibody, and do a very sensitive quantification. And now 6 labs in North America are doing this. It's really quite an exciting development. But remember, all of these are doing external calibration. All of these assays are using external calibration, and that's why we're getting good results from lab to lab. Okay, so to conclude this part, external calibration is superior to internal calibration. External, please, external. Use a sample in the digest to calibrate your sample, other than the sample itself. It reduces day-to-day variability within our own lab. It reduces system-to-system variability, and we're actually able now to standardize assays across labs, at least in the clinical arena. Please.
This is Scott, but thing that you showed before, so is the, should the internal standard be added at the digestion or after into the, where you're incubating the bees?
We've experimented a lot with that. We now add it before the digestion because we're getting, we're controlling for some of the digestion variability better. We're now digesting in about 2 hours, sorry, in about an hour, instead of overnight, which means a lot of that degradation doesn't happen anymore. If that makes sense.
Yeah.
But we still spike it early as we can. Good question. Yeah.
Could you discuss about assumptions here for the single-point calibration?
Assumptions for the single-point calibrator.
Because you have to have the assumption that the response is going to be linear.
You have to, yeah, absolutely. So you're not going to talk about parallelism and other studies like that, are you? Okay. To prove that a single-point calibrator is behaving similarly to other human samples, we can do mixing studies and parallelism studies. So by doing linear mixes between two, if we can prove that that's a linear relationship, if I can take purified protein and spike it into multiple human samples and assess the recovery that I get, that's the way that I try to prove that I don't have a matrix effect and that my single point calibrator is behaving like the rest of the samples in my batch. Does that make sense? So that those are some, I can point you in the direction of some papers to talk about how to do those experiments if you're interested. But so let me build on that question just a little bit. How should we calibrate tissue measurements, tumor measurements? Let me start there. I want to quantify a protein in breast tumor. What is my calibrator? What should I use? Should I use plasma? Probably not. Should I use bovine muscle? Probably not. Should I use normal human breast tissue? Maybe. Is there something else I could do? These are so getting back to what is the right matrix that I should be in takes some thought and takes some experimentation. The nice thing about having cell culture is if I'm going to measure proteins in cell culture to cell culture should be pretty similar, right? But that's not true if I'm using a cardiac myocyte culture versus like a CHO cell. They're not going to digest the same. They're not going to homogenize the same. So your question is an important one. Is the calibrator that I'm using really relevant? It's easy in plasma and serum. It's harder when we start to move into other places. But having something that you're digesting every single day will help. Other questions? It's back. Which part? The single point calibrator? We do. And my email is not on here, but I'll put up my email address at the end. Other questions? Okay. So now I'm going to go back to HDL Proteomics and tell you a basic science story. So this has nothing to do with Skyline. I'm sorry, but it's really fun data, so I wanted to tell the story. And it was the proteomics assay that got us to where we are today. So, as I mentioned, we're doing density gradient ultracentrifugation. We're making the HDL and we're digesting it into peptides. We're using an internal standard protein, Apolipoprotein A1, not internal standard peptides, and we're quantifying 38 proteins. So we have 2 peptides, sometimes 3, for each of the proteins, and we're using that internal standard, and then we're using a single point calibration approach to try to improve precision over time. We did this in one small study. So this is looking at folks who have type 1 diabetes. We're looking at coronary artery calcification, which is a measurement of atherosclerosis, at least as a surrogate for atherosclerosis. And we found that peroxidase 3 is actually reduced in patients with type 1 diabetes who have coronary artery calcification. And then we were actually able to replicate that in a different patient population, in lupus patients, we have a statistically significant decrease in peroxidase 3, which is one of these HDL proteins in lupus patients who have carotid artery plaque. I'm going to stop talking about peroxidase 3. It's something that we're still working on with collaborators at UW, but I don't have more story to tell you. But what was exciting about this is that peroxidase 3 appeared to be a marker in patients with autoimmune disease that had preclinical atherosclerosis. But then we wanted to take a look. I have very close collaborators in nephrology at UW, and we wanted to look at chronic kidney disease. Chronic kidney disease greatly increases the risk of having a myocardial infarction. There's more than 30 million Americans now with chronic kidney disease. Most of them will die from cardiovascular disease. They have way higher incidence of cardiovascular disease than the rest of the population. But LDL cholesterol doesn't predict bad outcomes in this population like it does in the general population. The thinking is that there's a big metabolic disturbance in chronic kidney disease. We're not able to clear out all those horrible things because our kidneys aren't working well. So we see increased amounts of reactive oxygen species, aldehydes, thiols. We also see increased inflammatory cytokines, and there's that systemic inflammation hypothesis that may drive cardiovascular disease and chronic kidney disease. In addition, the endothelial cell lining of our blood vessels is dysfunctional in CKD. And so the hypothesis was that HDL, because it protects the endothelium, by inhibiting activation of NF-kappa B, decreasing expression of cell surface markers, preventing inactivation of eNOS. The hypothesis is in chronic kidney disease, HDL doesn't work anymore. So can we use proteomics as a way to look inside the HDL and explain this? Previous studies have never really looked at, there's very severe chronic kidney disease, which is when you're on hemodialysis. And the previous studies had compared this very severe chronic kidney disease, and hemodialysis is pretty horrible to your blood vessels in general, versus perfectly normal people. And they had seen increased ApoA-I, which is serum amyloid A protein, which is an inflammatory marker, and ApoC3. And it appeared that HDL, the function was impaired with hemodialysis. In contrast to these previous studies, the Seattle kidney study, the stages of 1 to 5. So these are people walking around. They're not getting hemodialysis, but they do have significant chronic kidney disease, and we had lots of clinical covariance. I'm not going to talk about the hemodialysis fistula maturation study. So what do these people look like? They're 58. They're roughly 60 years old. They're more male than female. We have some racial diversity, not an extreme amount. The CRP is higher than what you see in the normal population. When we get chronic kidney disease, HDL cholesterol goes down. Interestingly, in the Seattle kidney study, it goes down a little bit, but it doesn't go down as much as it does in other studies. We don't know why. In addition, triglycerides generally go up. And part of the reason is that ApoC3, which is actually a regulator of lipoprotein lipase, is elevated. We should see the triglycerides going up, but they don't, not like we're supposed to. But when we look across this population, there was about 600 people in this SKF study that we did. So there's 600 proteomics measurements in these calculations. We can start to see that proteins begin to correlate with one another within HDL. So for instance, ApoA-I and ApoA-II, serum amyloid A1 and 2, correlate with each other. Apolipoprotein B and lipoprotein a, which are both very large atherogenic lipoprotein particles. PLTP and cholesterol across 600 people, the correlation coefficient was 0.9. It's almost like every time you have PLTP, you have cholesterol on the same particle, which was pretty interesting. We've replicated that over and over. There are some serum proteins which may be purified indiscriminately. Correlation between haptoglobin and haptoglobin-related proteins, and then the ApoCs, these little tiny regulatory proteins, seem to be clustering quite a lot. So we basically saw these correlations supported the previous biology that had been published in the past. So, given now all these data, we could ask which proteins are associated with the loss of glomerular filtration rate, or eGFR. And what we saw was that the worse your eGFR gets, the more ApoC3 you have, and the more retinol binding protein you have. Those are two statistically significant differences. But what we saw was a depletion of 3 other proteins, vitronectin, CETP, and ApoL1. ApoL1, so albuminuria is another problem that we have with chronic kidney disease. The loss of ApoL1 was the only statistically significant change that we saw with albuminuria. So when we fit this into a table, this is now the relative risk of having what happens to the proteins concentrations as we lose our GFR, as we get worse chronic kidney disease. So you can see an increase in RBP4, an increase in the ApoC3, but a reduction in the other three proteins. This is unadjusted. Because we had all those clinical covariates, we could adjust for age, race, sex, diabetes, BMI, etc. And you can see that we have statistically significant significant changes. But when we control for albuminuria, then the CETP falls out. So I'm only going to talk about the other four. And these are those data now plotted. So as we get worse and worse chronic kidney disease, we have an increase in two of the proteins and a decrease in the other two. So retinol binding protein 4 is a small protein. It's relatively abundant in serum, but the plasma concentration of retinol binding protein goes up in chronic kidney disease. So this is boring, right? So the HDL concentration of retinol binding protein goes up, but it goes up in plasma. So there's nothing new here. This is not very interesting at all. ApoC3 is a regulator of lipases. ApoC3 inhibits lipoprotein lipase, and the increase is what's thought to be associated with the hypertriglyceridemia that we see in chronic kidney disease. Again, ApoC3 goes up in chronic kidney disease. It goes up in HDL. Nothing very interesting. Vitronectin is it regulates just about everything. You can find vitronectin in every proteomics paper that you've ever read. You found it in ours too. We can do something to kidneys when we knock it out in mice, but nothing dramatic. But it is decreased in chronic kidney disease. It's decreased in HDL. It's decreased in plasma. Nothing interesting at all. But apolipoprotein L1, this was very exciting to us. It's a recent discovery, so it's only, is that 20 years ago? Is that 20 years ago? What year is it? Yeah, 20 years ago. Not that many publications about it. And it was originally identified as part of the trypanosome killing complex. So with haptoglobin-related protein, ApoA-I, this is what's protecting us from getting infected with trypanosomes. Pretty cool. It's fun in that ApoL1 inserts itself, it gets eaten up, the ApoL1 gets ingested by the parasite, then the ApoL1 forms a pore and causes the parasite to die a horrible death. And what we found, well, so first we had the hypothesis that because it's part of the trypanosome killing complex, ApoL1 and haptoglobin-related protein would be correlated with one another, but across chronic kidney disease, it actually was inversely related, which was surprising. We don't know why. But what we also figured out was that ApoL1 has different genetic polymorphisms. So as you look in descendants from different parts of Africa, their wild type we call G0, G0. Depending on where you're from in Africa, you may have a G1 polymorphism or a G2 polymorphism, concentrated mostly here in Western Africa. And it appears to be related to which trypanosome is most common in the different parts of Africa. These mutations, the G1 mutation and the G2, so the G1 polymorphism is actually two different changes in the protein structure. G2 is a single polymorphism, and this is the SRA interacting domain of ApoL1. This is what interacts with the trypanosome to help kill it. It turns out that the trypanosomes figured out how to interact with this part of the protein and prevent ApoL1 from working. Well, then we humans figured out a way to mutate our own ApoL1 to screw the trypanosome. Pretty fun. So the G1, G2 is actually protecting us from the trypanosome protecting itself, which is pretty cool. So that's probably why we see an increase in different genotypes in Western Africa based on what trypanosome is attacking. But what we also learned, so if you don't have any risk alleles, you die from trypanosomiasis. If you have one of these risk alleles, you can inhibit trypanosomiasis. But when you have two risk alleles, you actually have an increased risk for chronic kidney disease. And it's a significant risk. Sometimes in certain types of chronic kidney disease, having these two risk alleles gives you a 50-fold increased risk of having chronic kidney disease. So these are big mutations. What's interesting is you can tell the difference between G0, G1, and G2 from a single tryptic peptide, which is pretty fun. So we can quantify, we can actually genotype, we can quantify how much ApoL1 is, and we can genotype all in the same experiment. The genotype, ApoA-I, whether or not you have a risk allele versus a non-risk allele, will affect whether or not, if you receive a kidney from a donor who has a risk allele, that kidney is more likely to fail. There's been a lot of controversy about whether or not we should genotype kidneys before we give them to people. If we did that, we would actually reduce the number of kidneys in the donor pool, so we probably won't be doing that anytime soon, but that is one of the controversies that are out there right now. So ApoA-I is lower in HDL that I talked about as we get higher and higher worse and worse chronic kidney disease, but it's also significantly lower in blacks, and it's true all the way across. So these curves, one curve is fitting the white population, one is fitting the black population. We see a decrease, and it's constant, across the loss of kidney function, both in blacks and whites. But that doesn't make any sense. So, in 2016, a group from Merck published their quantification of ApoA-I using LC-MS in plasma. And what they showed was that blacks have higher concentrations of ApoA-I in their plasma, not lower. But I just showed you that ApoA-I is lower in ApoA-I is lower in the HDL from blacks, but in plasma, it's higher. They showed that looking at ApoA-I in plasma, there's no change as kidney disease gets worse. But we've shown now the ApoA-I in chronic kidney disease, the ApoA-I goes down as you get worse and worse kidney disease. So it doesn't match. That's pretty fun, right? We have biology now, something interesting. When you look at this plot, so this is the polymorphic peptide, so I showed you that this is the G0 peptide, this is one of the ones that we were quantifying in our assay. So if I have a mutation, I'm going to lose that peptide. If you look at a plot of the polymorphic peptide, if I have the mutation, I'm not going to find that. And I see that along the x-axis. The non-polymorphic peptide will be present both in G0, G1, and G2. But if you look at ApoA-I on the left and ApoE on the right, you can see there's a cloud here in ApoE. So these are the two polymorphic peptides. There's a polymorphic peptide in ApoE as well. And you can see a cloud here. And the cloud's not present in this ApoA-I. So again, we're learning something about the peptides in this proteomics assay. Please. We use density gradient ultracentrifugation to purify just the HDL fraction from plasma.
So all of the peptides directly binding to HDL?
Correct. Binding to HDL or making HDL or what have you, they are on the HDL particles in some way.
So if you were directly quantify the ApoA-I like is there any change?
So that's what these folks did at Merck. So when there's no change with chronic kidney disease. Yeah, it's the same. Does that make sense? Okay. So when I looked at this and said there's a cloud here, but there's no cloud here, I said to myself, I think because we know that this is the E2 null, this is the E2 heterozygotes, and these are the E2 homozygotes. We know that but where is the purple circle over here? Why isn't there a G1, G2 heterozygote on this figure? Maybe I was reading into it too much, but we decided to do some experiments. But before I go there, again, ApoA-I and HDL, polymorphic peptide versus non-polymorphic peptide. They published the same thing in their in their plasma paper, the polymorphic peptide versus the non-polymorphic peptide. And again, you can see this heterozygote cloud that we don't seem to have over here. And the R of 0.93 is matching their R of 0.91 in the homozygous G0, G0. So the hypothesis that we had was that the G1, G2 is not sticking to HDL, like the G0, G0. Biochemically, we don't know why, but that was our hypothesis that we thought we could answer with proteomics. So Michael Andrews is a collaborator at the CDC. They have a few of these instruments set up, this asymmetric flow, field flow fractionation. So you're able to take plasma, put it in, and then separate based on size using the flow of the countercurrent and with the current of the plasma. Have small things come out first, have large things come out last. So we're able to separate things based on size instead of density. this is pretty cool. The setup that they have is going from this AF4 asymmetric flow field flow fractionation, going on to a digestion. So they're doing inline digestion that's going straight onto the LCMS, which is kind of fun. So they're able to do that on their instruments. They also are looking at phospholipids and other lipids. So this is their protein, the Shimadzu inline digestion. I've never worked with that. Has anybody in this room worked with that? Inline digestion? No? Okay. It's coming to a theater near you. So, what I'm showing you here now is the stuff coming off of this AF4. The lower fraction number are smaller things, and the bigger fraction number are the bigger things. ApoA-I binds to IgM, one of the trypanosome killing complexes. That's what's coming out way out here. But this is G0 with normal. This is G0 with partially reduced kidney function, and this is G0 population with much lower kidney function. You can see that there's ApoA-I is different as we look at the different sizes of HDL particles. I'm now adding, it's a little bit busier, but I'm adding in now the G2, G2, and the G1, G1. And you can see that they're not here either. They're over here. So mutants and worse kidney function is coming out on larger particles than normal wild-type G0, G0 with normal kidney function. This is an even busier slide. This is what we're purifying in HDL. These are ApoE containing lipoprotein particles, which is apparently where ApoA-I likes to reside when you're at G2, G2, or have poor kidney function. ApoB, that's LDL, and as I said before, this is that IgM particle that's way out there. So it appears that there's actually a part that this is actually explaining what's going on. So we then quantified the shoulder, so we took the fractions that make up the shoulder, and we looked at each of the individuals that we had in this small study, and showed that if you are wild type but you have reduced kidney function, so this is a significant chronic kidney disease, you have less ApoL1 living in the HDL part that is the ApoA-I particles. You have normal GFR, you have more, and again, these mutant ApoL1 proteins that we learned from proteomics as biochemically behaving very different is now looking like they're living on different particles. So why is this important? My new hypothesis is that ApoL1 is literally making pores in the kidney, depending on urine pH, and causing toxicity in the kidney. And we're learning more about the biochemistry of that from the HDL. The relationship of ApoL1 to HDL is going to be similar to ApoL1 versus kidney membranes. This is supported by the difference between having one risk allele versus two, and whether or not this is whether or not ApoL1 is living as a monomer versus a dimer. It can't be a monomer, otherwise two risk alleles wouldn't be toxic, whereas we know that two risk alleles is toxic in human biology. So just to summarize the HDL part, these plasma concentration changes over eGFR match what we see in HDL. That's in contrast with ApoL1, which doesn't change across chronic kidney disease, but is clearly changing in the HDL. So the question is, ApoL1 changing density? Do they have a different affinity? Is there a connection with cytolysis? We don't know. So we've finished now genotyping SKS. We've done it all by proteomics. So again, looking specifically for those peptides, we can tell what the genotype is. No more PCR needed. We can do it in our own proteomics lab, which is great. And now we're looking for binding partners and other things, as well as intracellular regulation. So proteomics has taken us quite far. The other thing that I wanted to talk about, so we've taken all of these HDLs and we've also looked in lipidomics, and I just wanted to give a shout out to Skyline. You can do lipidomics with Skyline as well, which has been awesome. It's made data analysis so much easier. And now, just looking again across these 600 people at many different lipids, phosphatidylcholine, these are glucosylceramides, lactosylceramides, sphingomyelins. We're able to look at all of those at once, and we can start to see patterns of these lipids as well. But I don't have any data looking at outcomes for you today. Lots of people to thank. I have amazing collaborators, Ian and Brian. I mentioned Jess, one of the most talented technologists that I've ever had the pleasure of working with. The IGF-1 working group is around the world, and again, that assay is now being used for anti-doping in sports. I talked about the collaboration with the clinical labs around the country, and the most recent data from the CDC. Michael Andrews is a burgeoning scientist and really fun to work with, and lots of help from funders. So thanks to you guys for listening. I think we have time for questions, I think. Yes. Questions, please.
Have you looked at any like gut microbiome stuff in association with some of the things that you're seeing? Some of the cyanobacterial toxins are known to be hepatotoxic.
We've only looked in mice. We haven't looked in people yet. So a collaborator, Karin Bornfeld and Jenny Cantor at UW, showed that it appears that gut permeability, or the ability of a bacterium to cross the gut barrier, is correlated to atherosclerosis in mice that have type 1 diabetes. So the hypothesis was that there would be specific microbes that are crossing the gut that would lead to increased atherosclerosis in mice. And it turned out that that wasn't true. So that's the only thing that I've seen so far. I haven't paid close enough attention to other gut microbiome data to see if there's an interaction with the HDL cholesterol question. I haven't looked at that. It's a good question though. Love it. I mean, your gut is, you are your gut, for sure. Yeah.
Have you looked at integrating the lipidomic assay with the proteomic results?
We have. It's very complicated. It's bizarre. No lipids correlate with proteins, which I was not expecting. I thought that there would be a lipid pattern that would kind of cluster with a protein pattern, but that wasn't true. The lipids and the proteins almost appear to be behaving completely independently of one another. Yeah, but no, I would love to say that there's a good story there, but I don't have it.
How do you assess the stability of your single-point calibrator?
How do we assess the stability of the... Yeah, so in the clinical world, we have proficiency testing. So proficiency testing means that someone else is looking at me and telling me how good I'm doing. So for thyroglobulin, there's a proficiency testing material comes in, we can analyze it, we send the results back, and then they tell us whether or not we have the right concentration. So that's the drift in the assay. If the calibrator weren't stable, the concentration that we would be getting in our assay would drift as well. So I guess what my answer to your question is, we have external processes to prove that our calibration is stable over time. How would you prove that your calibrator in your own lab is stable if you don't have anybody else to compare with? And that's where having a quality, which I didn't go into, having quality control material that we can analyze repeatedly over many days, over many weeks, over many years, with different calibration materials, you should get the same answer every time. So yes, quality control materials is really the best way to show that your calibration is stable over time.
Yeah, I guess the real challenge is to have another assay.
Yeah.
To see that.
You can do quality control in your own lab though, right? If you have quality control materials that you can run with every batch and show that that concentration doesn't change, you can be comfortable that the results for your unknowns is accurate. That's the basis for doing QC. I have some slides on quality control if you want me to talk about it.
Are you talking about recombinant or endogenous?
Well, it depends on what the calibration system is, right? For the single-point calibrator, everything's native. I'm not spiking anything in, right? I don't want to spike anything in. I want it to be the native protein.
The native protein, the recombinant source of native protein, you're talking about spiking like an endogenous.
Oh, when you're spiking stuff in, if you're going to spike stuff in, you will buy one of two varieties. One is purified, right? So you can buy retinol binding protein 4, for instance. You can buy a purified version of that protein that you can then spike in. It's purified from urine, so whether or not it's different. Or you can buy recombinant retinol binding protein 4, significantly less expensive, but it also lacks some of the post-translational modifications that you get from the RBP4 that's been purified. So we got both, just to ask the question, if I spike purified versus recombinant in, do I get the same slope? And the answer is yes, so that made us feel good. Yeah, but the stability of the calibrator is a great question, but quality control is my only answer.